All right, you guys. Pretty good turnout for just announcing this today. Let's see here. While I'm still setting up, does anyone have a burning question right up front? Not yet. Okay, I'm just going to bring my computer up here. Let's see. Bring the screen down. All right. Do you guys just want to start? That sound okay? All right. Let's just start with GI physiology. You guys, this is really informal. Ask questions as soon as they arise, as soon as they come up. I'll just go through and kind of tell you guys what's important, what's not important. Like the teeth, not going to be on your exam. So we'll go through that, but I mean, we won't go through that now. But I'll just tell you like what's going to be on the exam, what's not going to be on the exam. All right. So, Anita. Yeah, absolutely. Dim the lights. All right, let's see here. Oh, that's the opposite. Whoa. There we go. Oh. Everything but. What are these like right here? That's better, isn't it? All right. I'm sure the people at home that are going to be listening to this are going to be going, oh, what's going on? All right. Okay, so remember the GI tract, you guys, is made up, it is an organ system. It's made up of a lot of different organs that all work together towards a similar function or set of functions. And what I'm talking about is digestion and absorption. Okay? Digestion and absorption. Mainly. There are 4 different processes, actually. They include digestion, absorption, secretion, and motility. Okay? So that's the major things. We kind of talked about just an overall introduction of all of these different organs. One by one, we'll actually be going through the main organs that make up the GI tract are the upper GI tract. We'll talk a little bit about the mouth. The salivary gland is the most important part when we're talking about the upper GI. Then we'll talk about the esophagus and some of the muscles that control the esophagus. I think that's the major thing to know about the esophagus. And peristalsis. Remember I told you guys the story of my daughter and swallowed the quarter? So that gave you an idea of peristalsis and secondary peristalsis in the esophagus. Then the stomach. You can actually see the stomach. The stomach, there is a number of cell types that are important. We'll revisit the parietal cells, the chief cells, G cells. We'll talk about enterochromaffin-like cells. Those are the ones that secrete histamine. And the trifecta is gastrin, acetylcholine, and histamine. So we'll review that. So the stomach is all about producing and secreting hydrochloric acid, helping with chemical digestion, and then there's mechanical digestion as well. Moving on from the stomach is the small intestines. We didn't really break it down for our purposes. We just kind of overall talked about the small intestines. But the upper part of the small intestines is called the duodenum, or some people pronounce it duodenum. I'll know exactly what you say, whether you pronounce it one way or the other. I'll know what you mean. And then the jejunum, and then the ileum is the last part of the small intestines. Then we'll go through the colon. The main thing to think about when we're going through the small intestines and the colon, the lectures are kind of broken up into motility. So you need to know the motility of the small intestines and motility of the large intestines. So I'll just review that overall. We can go through this again. But the motility in the small intestines is segmentation, again peristalsis, and then what's called MMCs, or migrating myoelectric complex. Okay. In the large intestines, we talked about hostration, and we talked about mass movements. And then what I need you guys to think about is overall function. We spent a lot of time in the small intestines talking about digestive enzymes and how they break down carbohydrates, proteins, lipids, and then a little bit about RNAs. How RNases and nucleotidases actually break down nucleotides. Okay, and then the large intestines, their function is really about forming feces and moving it down the line. There is some absorption in the colon, but it's really about making feces. And then finally the rectum and the anus, and that's all considered the alimentary canal. So another thing I guarantee will be on your exam is the defecation reflex. So we'll go ahead and review that as well. Okay, so that's just an overview of what we're going to kind of cover tonight. Just remember that there is an enormous amount of fluid secretion and absorption that occurs in the GI tract. And I always like to talk about that up front. I want you guys to think about that when you think about transport. Only because, and this is interesting, not a lot of aquaporin channels have been characterized in the small intestines. And look how much fluid is absorbed in the small intestines. It's known now that a lot of water is absorbed by the sodium glucose transporters, those SGLT. That's why oral rehydration therapy, you need to include electrolytes like sodium and glucose to be able to rehydrate someone. You can't just give them pure water. They won't absorb it. A lot of it will just run right through them. So anyway, you can kind of look this over. You don't need to know specific amounts that are coming from the salivary secretions. Okay. Again, the GI tract is organized in compartments that are usually gated by sphincters. True sphincters are made up of circular smooth muscle that constrict when they contract. And what's really cool about the GI tract is these compartments stay fairly separated by these sphincters. I think it is amazing that the stomach, after you eat a meal, can secrete so much acid that the pH falls to 1 to 2. And that doesn't destroy not only the stomach, but all the compartments around it. It is amazing, I think. Okay, so compartments can vary in pH, enzyme composition, and all types of secretive and absorptive cells. Okay? All right, so I'm going to say just skip the teeth. Unless you want to become a dentist. Isn't that Rudolph? What is it? What's the cartoon? Is it Rudolph? Yeah. Yeah, with the little guy that wants to be a dentist. The little elf, right? Unless you want to be that guy, don't worry about the teeth today. All right, but the salivary glands are important. Okay, so do really think about the different salivary glands. The parotid gland is the largest gland, but at rest, it only contributes to about 25% of salivary secretions. when you eat a meal, it actually bumps up from 25% to about 50%. So I would know where the parotid gland is. It's kind of near the upper mandible. Kind of see this on the figure here. The main thing to remember is that the parotid gland secretes a very serous fluid. What that means is it's very watery. Serous fluid. And it contains amylase. So already you have enzymes that are being secreted by the parotid gland, the amylase that is starting to break down carbohydrates in the mouth. Cool. The next one I like to talk about is the sublingual. Let's just skip to this third one because this is the other extreme. This one is right under the tongue. Oops, sorry you guys. Here's the parotid gland. I thought that was showing. So here's the parotid gland. And that one is the largest. Again, it secretes a very serous fluid. And then underneath the tongue is the sublingual gland. And that one is the one that it really only secretes about 3 to 5% of salivary fluids, but it's rich in mucins. Produces a very thick mucus. Like I said, I always think of St. Bernard's or my dog when he's watching me eat. He's got those long, long salivary secretions that are just kind of hanging there. And I have to tell him, what's the matter with you, Hank? Pull yourself together. Pull yourself together. Okay, so that's the sublingual gland. It really secretes a very mucins, lots of mucins. The submandibular gland right here, all you need to remember is that's kind of intermediate between the parotid gland and the sublingual gland. The submandibular. So why is saliva important? It helps to lubricate food. It already secretes amylase to start digesting polysaccharides. Let me flip to this next one here. Saliva lubricates food, digests polysaccharides by secreting amylase. Dissolves food so nutrients can bind to taste receptors. And then it already secretes antimicrobial agents to start to eliminate bacteria in that area. So remember that these glands are true what's called acinar glands. Acinar glands. So I don't know if you guys remember this when we actually talked about it during the epithelial transport section, but this is what's called an exocrine gland because the secretions are delivered to the outside of your body or a compartment. Okay, so like a sweat gland would secrete fluid to the outside of your body. But salivary glands, they secrete fluid to the inside of your mouth. Basically, that's still kind of the outside world. Yeah. So acinar glands, they're divided. They have two parts. They have an acinus. This is the bulb-like part. And then this tube is the duct. So it has two parts, the acinus and the duct. Okay. So this figure is a little bit cryptic. This symbol right here is the capillary beds. These are the capillary beds. Fluid travels from the blood, from the capillaries, and is transported into the lumen of the acinus, into the middle region here, okay? So that means that these primary secretions here are very plasma-like. That's very plasma-like. because the forces, you guys remember Starling forces, it's really just about physics, dictates that the fluid's going to move from the capillaries under high pressure, and they're going to be forced into the lumen of the acinus. So when asked what is the composition of that primary secretion, you can say it's very plasma-like. Very plasma-like. So then these primary secretions then are forced into the duct portion of the salivary gland. The flow is actually controlled by these myoepithelial cells. So when they constrict, they actually decrease the flow, or they dilate to allow a greater flow. But you can actually see here that there is a significant amount of sodium chloride reabsorption, so that you don't lose a lot of those electrolytes to your environment. Now, these nerves, these are the neurons here, they are also regulating the flow, but also the amount of secretion. So the parasympathetic system, that resting and digesting, is responsible. You can think about this acronym. This isn't going to be on your exam, but SLUD. That's connected with the parasympathetic nervous system. It's secretions, namely secretions like salivary glands. So the parasympathetic increases secretions, lacrimation, which is tears, urination, and defecation. So when you think about parasympathetic, you can also think about the acronym SLUD. Okay, so secretions is part of this. The parasympathetic actually increases the amount of secretion, and then you can see that it is delivered to the outside of the mouth. Okay? That's about it for the salivary glands. Okay, taste receptors, cool stuff. I talked in both classes about the taste receptors in the gut. I think that's a really cool story. Just remember that those taste receptors detect glucose, but also even artificial sugars. And they respond by increasing the number of sodium glucose transporters. So that even in the face of Diet Coke, whatever you eat with that Diet Coke, you're going to be able to absorb more sugar into your blood. So that's the only thing that, remember, we did talk about the different 5 taste receptors too. Yeah, you have a question.
You're talking about the taste receptors in the gut?
In the gut.
Okay.
Yeah, in the small intestine. But let's go back just to review. The taste receptors are sweet, salty, sour, bitter, and umami, my favorite. Umami. Umami taste receptors detect amino acids in meats, like protein. Monosodium glutamate, that will trigger a very specific umami receptor. Then you have the sweet taste receptors. Sour is really just detecting acid. Okay. Bitter, you've evolved bitter taste receptors so that you can avoid poisons. And then salty, just detecting sodium. Okay, cool. Yeah.
Does it increase the sodium glucose transporters?
Why does it do that? Oh, where does it? In the small intestines, the duodenum. Okay, so the swallowing reflex is also an important, it's all mediated by the medulla oblongata. Okay, so it's a coordination between skeletal muscles, sphincters, and smooth muscle groups. And the thing to remember with swallowing, this is called deglutition, is that there's 2 phases. There's a voluntary phase and there's an involuntary phase. So the voluntary phase goes like this. You take your tongue and you push the food to the back of your throat, the pharynx. The pharynx is basically the back of your throat here. And then before you actually swallow, there's muscles that keep your esophagus closed. Then the involuntary phase takes over. That epiglottis, that omega-shaped structure, bends over the windpipe, bends over the trachea. I should say this is the larynx area, glottis larynx. And the soft palate moves up to protect your nasal cavities so that you don't get food up your nose, basically. Unless you laugh, like I said, while you're drinking milk, you're going to get it in your nose. All right, but usually the soft palate kind of lifts up, rises up to close the passage to the nasal cavity. Epiglottis bends down to close off the trachea. All right, so the thing to remember, I can tell you there'll be a question about this too. The thing to remember about the esophagus is that it's under different muscle group control. That's not a good way to say it. There's two types of muscles that are controlling the movement of food from the mouth to the stomach. And really, to be honest, the esophagus really is just a tube. It's a tube that transports food from A to B. That's pretty much it. There's no absorption, there's no secretion. It's really just motility, movement, peristalsis that moves through food from A to B. Now, the thing to remember, and I think this is something you might want to star, you can see here, a lot of students kind of miss this. The upper part of the esophagus is under skeletal muscle control. The bottom part of the esophagus is under smooth muscle control. The reason why I say that is because myasthenia gravis, right? This is a very important way to diagnose animals with myasthenia gravis. What usually happens, again, it causes profound muscle weakness of particularly skeletal muscle, okay? Because antibodies attack the nicotinic acetylcholine receptor. That's the receptor that acetylcholine binds to cause muscle contraction, okay? So when you get antibodies that attack the nicotinic acetylcholine receptor, it causes profound muscle weakness. And in dogs that have myasthenia gravis, the way they diagnose it is they can actually, when they image the esophagus of these dogs, they can see what's called megaesophagus. It causes muscle weakness in this particular area. So this larger part, this upper part of the esophagus is much larger, and sometimes food can actually accumulate up there. It has a hard time pushing the food down into the stomach. Again, they can image those dogs and they can see it, and that's one way to diagnose patients with myasthenia gravis. Okay, so what you'll notice, the other thing is that's important in this slide, is that the upper esophageal sphincter and the lower esophageal sphincter start off not with a pressure of 0, but you can already see that there's a basal tone associated with these sphincters, this circular smooth muscle. You guys know what I mean by that? Okay, so the circular smooth muscle at the top of the esophagus, called the upper esophageal sphincter, and the lower esophageal sphincter, they're already constricted, okay? They're already tightened up. So if I took some kind of apparatus that could measure force or tension, I could already measure the pressure in the inside of the wall associated with that constriction, okay? So when a bolus of food actually enters into the area right above the upper esophageal sphincter, you can see that there's already a relaxation that occurs, and then that bolus of food moves through. So those sphincters have to relax to allow for food to move through that area. And for the upper esophageal sphincter, it starts to relax once a bolus of food, a big wad of food, enters right here. Then the upper esophageal sphincter relaxes. That's this dip in pressure. And then when that food moves through that area, you can see an increase in the pressure again. Now, I'm going to skip all the way down to the lower esophageal sphincter. And I want you guys to notice that when a bolus of food kind of passes right across this upper esophageal sphincter, there's already an anticipatory relaxation all the way down here. See where this arrow is? This is what's called an anticipatory relaxation, even before food is even close to the lower esophageal sphincter. You can see this bump right here is the food traveling down the esophagus, and it doesn't reach the lower esophageal sphincter for a number of seconds. But you can see that there's, again, an anticipatory relaxation. Okay? So remember that motility is usually peristalsis. Peristalsis in the esophagus is when circular smooth muscle constricts behind that bolus of food, and then it shortens the longitudinal muscle to literally like push it down the esophagus. That's my sweatpants analogy. Yeah. So peristalsis, if you have a big bolus of food, peristalsis is when there's circular smooth muscle that constricts behind that food. And then the longitudinal muscle shortens, so that what happens is it tends to push the food down the line. And it's very rhythmic. Like I said with my daughter, when she swallowed that quarter, she was clutching her chest and then relaxing, and clutching her chest and relaxing. I mean, it was very rhythmic. So it's kind of interesting. I fall short of saying, that was really cool. Sorry. All right, so that's the main thing to understand about the sphincters and about the muscle control. And the motility is called peristalsis. You guys have any questions about that? Pretty straightforward. I think the number one thing that students kind of mix up is the skeletal muscle versus smooth muscle. I've had a lot of interesting questions about that too. Like students come and say, oh, I thought skeletal muscle is all voluntary control. Why can't I control that? Good question. I don't know. I think maybe sometimes those sword swallowers, I think they might have control of that. I haven't counted that out. But good question. I can't really answer that. But that's skeletal muscle is the upper part, smooth muscle below. All right, so now let's concentrate on the lower esophageal sphincter. There's two different diseases that I want you guys to know for sure. We'll start off with esophagitis. Esophagitis is a condition where the lower esophageal sphincter becomes inflamed because it's too relaxed. So it loses its tone. And when it becomes too relaxed, it is not a great gateway. Now it's allowing stomach acid to gurgle up into the esophagus, which really can destroy those cells. Not only does it cause a really horrible pain, but if you leave that too long in your life, if you have chronic heartburn, it can lead to esophageal cancer. A lot of people that drink, that sometimes happens, leads to esophageal cancer. So initially, this esophagitis, anything like I said, itis at the end is an inflammation of the esophagus. All right, so what is achalasia? Achalasia, I think about this as kind of the next step. If you have chronic esophagitis, sometimes that leads to so much scar tissue that now the lower esophageal sphincter fails to relax. This actually happened to my uncle. He, again, allowed heartburn to go on way too long. And when that happens, you get food stuck right here, right at the top of the lower esophageal sphincter. He literally had to go into the emergency room to be tubed to get the rest of that into his stomach. Once it's in the stomach, it'll pass. But when it's stuck in the esophagus, it's very uncomfortable. Okay? All right. Don't worry about the different mediators. ATP and VIP tend to cause relaxation, but that won't be on your exam. So I don't want you to have to worry too much. Don't worry about these neurotransmitters. Okay, so that won't be on your exam. The vagus nerve, just remember the vagus nerve in general. I'm just talking in general. The vagus nerve is very important in the GI tract everywhere. And it secretes, like all parasympathetic neurons, acetylcholine. So remember when we get to the stomach, the vagus nerve is really important. Long, long time ago, people used to be treated to treat heartburn. They used to do a vagotomy. Vagotomy. Cut the vagus nerve. Seems harsh. Then all of those functions, urination, defecation, all of that go away. I mean, make it harder. So, yeah, that's not a good idea, but that's what they used to do, vagotomy. Okay, let's move on to the stomach. I think the bulk of a lot of the questions on the exam are from the stomach and the small intestines. Okay? So, there is a lot of regulation. The stomach is basically divided up into different parts. The fundus is at the top here. The middle of the stomach is called the body. And then the bottom part closer to the pyloric sphincter, I don't call that a sphincter, I actually call it the pyloric junction, is the antrum. Fundus, body of the stomach, and antrum is the lower part of the stomach. Okay, so the top part of the stomach up here, that's where receptive relaxation occurs. Receptive relaxation. That's an important term to understand. With receptive relaxation, I'll go to this next slide. You actually have mechanoreceptors. That's what these combs are. These are mechanoreceptors. These mechanoreceptors detect stretch or distension. I get a lot of questions during the exam. What is distension? Distension is just stretch of that area. It triggers those mechanoreceptors. So you get stretch of the fundus when you start to eat food. That causes, you can see there's a response. It sends a signal to the central nervous system by the vagal afferents, all the way back to the stomach by the vagal efferents. And that causes relaxation of the fundus. That's called receptive relaxation. The relaxation occurs so that the stomach can accommodate more food coming down the line. And then, yeah. Okay, so it's initiated by stretch. So as soon as you eat a little bit of food, there's going to be a certain amount of stretch in the fundus. That's going to send a signal to the central nervous system, all the way to the brain by the vagal afferents. And then the brain sends a signal back to the stomach by the vagal efferents. A lot of times it's mediated through this VIP. It actually activates a potassium channel that hyperpolarizes the cell. You don't need to know all that. But what you do need to know is the end result is a relaxation of the fundus so that the stomach can accommodate more food. It's assuming that you're going to be eating more stuff. Okay. Peristalsis. So what we'll learn in just a minute is that after you start eating food, you're going to get maximum hydrochloric acid secretion. There's going to be maximum motility in the gut or in the stomach. It actually becomes a very violent place. There's a lot of mechanical digestion. And then as soon as a little bit of food at this point gets kicked by the pyloric junction, there's a constriction, an immediate constriction to kick all that food back. This is part of the mixing process. Mixing and churning. And again, it's a pretty violent place. Okay, we did talk about the vomiting reflex too. So this is just kind of intro to stomach physiology. The vomiting reflex is mediated through the vomiting center. You actually have a vomiting center. A vomiting center. Oh, I don't think this tells you where it is. I believe this is in the medulla oblongata as well. But this is all about pressure changes. It's different than regurgitation. What happens is you start to retch. You breathe in against a closed glottis, like that, retching. And then it creates a negative pressure in the upper esophagus. At the same time, your diaphragm and your abdominal muscles are contracting to increase pressure in your stomach, creating a massive pressure change between your stomach and the upper esophagus. And then once that lower esophageal sphincter opens, that's when you vomit. Different than regurgitation. So whenever you hear about birds regurgitating, they are not vomiting. They're actually, regurgitation is usually reverse peristalsis. Same mechanism. Circular muscle constricts behind the regurgitated food, and then longitudinal muscle shortens, pushing that food up to the mouth. Okay, so let's kind of now concentrate on the cells of the stomach. Okay, so we're zooming into the wall of the stomach. We're going to talk about these different cell types. The main ones that I want you guys to know are the parietal cells and chief cells. Those are the two main ones. Now, what protects these cells from the high acid or low pH in the stomach? These cells are connected together by very tight junctions so that the acid doesn't leak in between the cells. And these mucous neck cells are secreting a lot of bicarbonate-rich mucus, which line the top of the mucosa. So it really does protect the wall of the stomach. Okay, so tight junctions, those mucous neck cells are secreting bicarbonate-rich mucus, and that protects the parietal cells and the chief cells. The endocrine cells, like the G cells, they secrete hormones. So, kind of looking at the different layers, the mucosa, that's the layer that basically faces the inside of the stomach, made up of epithelial cells in gastric pits. That actually helps to increase surface area. So you can see these gastric pits. Let's go to this next one. You can see how they're organized into the mucous neck cells, the chief cells, and the parietal cells here. Parietal cells here are the big orange ones. The chief cells are these pinkish ones, and the blue cells right here are the mucous neck cells. All organized in these gastric pits. So I'm going to go back. The mucosa has the epithelial cells, the submucosa underneath. That also has a layer of muscle. And then below that, nope, that's the mucosa. Below that is longitudinal muscle and circular muscle. And then this would be where the blood supply is down here. All right. So let's kind of talk about what parietal cells do and what chief cells do. The main idea here is the parietal cells secrete acid, but they also secrete one other molecule that I want you guys to know called intrinsic factor. Okay, so maybe I should I write this on the board? Intrinsic factor is a molecule that helps with the absorption of B12, vitamin B12. And B12 is crucial in the synthesis of red blood cells. So if you don't have intrinsic factor, you don't get B12 absorption, you get anemia. And it's a specific type of anemia called pernicious anemia. Pernicious anemia. I think it's P-E-R-N-I-C-I-O-U-S, pernicious. So intrinsic factor is actually essential for life. It's one of those molecules that is essential for life. Now that acid is responsible for actually converting pepsinogen to pepsin. Now let's go back to the chief cells. Chief cells secrete the precursor pepsinogen. Acid converts pepsinogen to pepsin, and pepsin is an enzyme that breaks down proteins to polypeptides. So you're already getting a significant amount of breakdown of proteins in the stomach. I would say larger proteins, because at this point those proteins, like a hamburger that you just ate, is still in solid form. So the pepsin actually starts to break down those bigger peptides or bigger proteins into polypeptides. And then it's really, you've got trypsin and chymotrypsin in the small intestines that is breaking it down further. Yep. Okay, so zooming in on the parietal cells now, I think this is where these sets of slides now, I don't want you guys to get confused. We're going to zoom in on the parietal cells specifically, and then talk about the regulation of parietal cells by those ECL cells, entero-like chromaffin cells, okay? So, parietal cells, this is one big parietal cell. Okay, so I'm just kind of zooming in on, right, here's our gastric pit, here's the parietal cells. We're zooming in on this particular parietal cell. And we're looking at the transport properties. Now, you guys have seen this equation 1,000 times before. We actually saw it in the kidney. We talked about epithelial cells of the kidney. We saw it in red blood cells, right? And the hamburger effect, carbon dioxide transport from the tissue to the lungs and back. So here we see it again in these epithelial cells that line the stomach. Carbon dioxide and water is converted to carbonic acid by the enzyme carbonic anhydrase, where then it actually produces bicarbonate and protons. Now again, this is the luminal side. This is the apical membrane. So this side faces the inside of the stomach. This side faces the basal, this is the basolateral side. It faces the blood supply. So for every carbon dioxide molecule, one proton is secreted and one bicarbonate is absorbed into the blood. Now let's follow this proton first. This proton is actually secreted into the stomach by this proton pump. This proton pump uses ATP to move protons against their concentration gradient and potassium. Okay, so potassium also is brought into the cell by this proton pump. Again, the purple pill, Nexium, the actual not the commercial name, but the generic name is called omeprazole. Omeprazole. In an exam, on the questions on the exam, it will say something like the proton pump omeprazole. Okay, so don't feel like you have to know omeprazole. I'll kind of put it into context for you. Okay, so omeprazole is the proton pump inhibitor. And this potassium channel is really important in making sure that potassium doesn't become rate limiting and inhibit the pump. So it allows for a little bit of potassium to exit the cell so that it can recycle and the pump won't become rate limited. And then because of the protons and the potassium, the net movement is cations into the lumen of the stomach. Chloride tends to follow. Okay, so literally, I'm saying you are secreting hydrochloric acid when you eat a meal. Now, on the flip side here, bicarbonate is transported into the blood by the chloride bicarbonate exchanger. This is an exchanger. Chloride bicarbonate exchanger transports bicarbonate into the blood. After you eat a meal, this is actually called the alkaline tide. For some reason, I always think of Alabama. Why? Roll Tide. Yeah, that's why. That's why I must think about that. It's the alkaline tide. So when you throw up all that acid, but you've just delivered a ton of new bicarbonate into the blood. You can become very highly, profoundly alkalotic as a result. That would be a metabolic alkalosis. Okay, so let's talk about the regulation now of the parietal cells. Regulation. Regulation of parietal cells. You have hormones, paracrine signaling molecules, and neurotransmitters that are actually playing a role in regulating the amount of acid that's produced by these parietal cells. They all have effects by inserting those pumps, the proton potassium ATPase, into the apical membrane of these cells. So you have one inhibitory, somatostatin. Anybody remember what else somatostatin inhibits? Talked about it today. Growth hormone, release of growth hormone. It also inhibits insulin and glucagon. Whenever you think about somatostatin, I would just think it inhibits a lot of stuff. All right, so let's talk about the regulation of these three. And remember, what they do is ultimately they insert those proton pumps into the apical membrane so that you get more acid secretion. So here's the three, the trifecta. This happens during the gastric phase. Okay, so at first, when you just start eating a meal, you get a little bit of acetylcholine release and a little bit of gastrin release from those G cells in the stomach. Both of those individually have effects on increasing acid production and secretion into the lumen of the stomach. But then when you start ramping up the system, you start eating more and more food, you can see acetylcholine and gastrin also trigger the ECL cells that are nearby to secrete histamine. And this is a different pathway. You'll notice that acetylcholine and gastrin are GQ coupled, but histamine, if you remember, cyclic AMP, is GS coupled. So now you've just brought on a brand new pathway. And with all three of these on board, you get the maximum amount of hydrochloric acid secretion. Maximum. This is like the trifecta. Yes?
Is it just histamine that's GS coupled? Or?
Yep, just histamine. Yep. Yeah, the other two are GQ. Yep. Okay, so this really helps to ramp up the system. Now, I do want you guys to know what potentiation means. Potentiation means that the amount of acid secretion is greater than the sum of the individual parts. That's a hard one. The potentiation means that the amount of acid secretion is greater than the individual parts. So let me just show you. Acetylcholine, all by itself, produces a little bit of acid. Gastrin, all by itself, just with gastrin, if there's nothing else on board, it produces a little bit of acid. Same with histamine. If acetylcholine and gastrin weren't there, it would produce a little bit of acid. Okay? If you add all these up, it still doesn't reach the amount of hydrochloric acid secretion that happens when all three are on board and potentiation happens. So potentiation means that the amount of acid secretion is greater than the sum of just acetylcholine by itself, just histamine by itself, just gastrin by itself. Potentiation means you're getting more. It's greater than the sum of the individual parts.
So
Where's the rest of the acid coming from?
Yeah, that makes it more.
Okay. The reason why you're able to secrete more acid is because you're actually inserting more of these pumps. Yeah, good question. It seems like when all pathways are on board, you get more pump insertion than just if you have just acetylcholine or just gastrin or just histamine. Yes.
So is it kind of like summation?
Summation. It's not just summation. So if let's just say that acetylcholine inserts 10 pumps, gastrin inserts just all by itself 10 pumps, and histamine 10 pumps. When you have all three on board, you get 60 pumps, not 30. You're actually inserting way more than the sum of the individual parts. Does that make more sense? Okay, so that's what potentiation means. I'm going to use that because I always get those funny looks like, what does that mean? You actually have to put some numbers to it, I think. Yeah, that makes more sense. Okay, so the rest of this is really just kind of flow charts. Now this is just about putting some things together, okay? The main idea here is to understand the difference between the cephalic phase, the gastric phase, and the intestinal, or what's also called the enteric phase. There's three phases: cephalic, gastric, or intestinal phase. Okay, so this kind of sums some of it up. The cephalic phase, you haven't eaten anything yet, nothing. But your brain can smell stuff. Your brain can see stuff that's being on the stove. So it's all mediated through these vagal efferents. It's all coming from brain. You're already getting some acid secretion through the parietal cells because of the acetylcholine that's released through these vagal efferents. The vagal efferents also trigger the release of what's called GRP, or gastrin-releasing peptide. That triggers those G cells right here to release gastrin. Remember, that's a true hormone, so it has to enter into the blood and then feed back to the parietal cells. So you're already getting a certain amount of acid secretion. But then once you eat something, here's the gastric phase. Remember these combs. Can you see that right here? These comb-looking structures, those are the mechanoreceptors. They're detecting stretch or distension. So now you're getting stretch of these areas, and that feeds back all the way to the central nervous system and back. Those are called the long reflex pathways. But it also kind of feeds back to these short little reflex pathways in the stomach. Here's a mechanoreceptor. It's actually just releasing neurotransmitter right here. and it's feeding back to the parietal cells. This short little neuron is actually releasing acetylcholine. Okay, so during the gastric phase, that's where you start getting the ECL cells, those entero-like chromaffin cells that are releasing histamine. During the gastric phase is where you really start ramping things up and getting maximum acid secretion. So here's a nice table, kind of giving you an idea of what happens in the cephalic phase, gastric phase, and now the intestinal phase. Gives you an idea that acid secretion, more acid secretion by long and short neural reflexes. Finally, we get to the intestinal phase, okay? So again, this is kind of going through that. I'm going to end up right here, okay? Okay, I forgot to mention some of the motility in the stomach. Remember, the stomach actually has 3 different types of muscle. Circular smooth muscle constrict. Longitudinal muscle shorten the stomach. But there's also these obliques that actually twist the stomach as well. So that's ramping up motility. When you're in the gastric phase, you've got the maximum motility going, maximum acid secretion. You're really digesting that food. You got pepsin on board. You're digesting that food mechanically and chemically, okay? Then all of a sudden, you move into the intestinal phase. You get a little bit of food into the intestines, everything slows down. That changes everything. Now you get a decrease in gastric emptying. That means the motility slows down, acid secretion slows down. Emptying means the food coming from the stomach into the small intestine. You get a decrease in gastric emptying. Seems a little counterintuitive, but now it's not delivering that whole wad of food from the stomach into the small intestines. It's not about that. Now it's about slowing everything down and piecemealing the food into the small intestines, little by little, so that you can maximize digestion and absorption. So it has effects on decreasing gastric emptying. You can see that there's local, through the nerves, through the submucosal plexus, they kind of increase pyloric contractions, decreasing gastric emptying. You get a decrease in the intensity and frequency of the peristaltic contractions and a decrease in acid production. That's kind of all labeled right here. Decrease in acid secretion for verbal learners here. And now you start to decrease gastric emptying as well. Okay, so let's go back to our chart here. Here's where you get all that hormonal regulation, and there are a lot of questions pertaining to this. I'm just putting it out there. Lots of questions on the hormonal regulation during the intestinal phase. Okay, so heads up on that. Great regulation, though. I think this is a really elegant system. This is one of the best stories I think the GI tract has. So you get a little bit of fat that's delivered into the small intestines from the stomach, and it's detected by eye cells. Okay, those eye cells detect lipids. Anybody remember the hormone that's secreted by the eye cells? CCK, cholecystokinin. Remember, you just have to recognize that on the exam. So CCK is released by the eye cells. It's a hormone, so it travels to the pancreas and the gallbladder. Does a few things. It stimulates the gallbladder to contract, and it stimulates the pancreas to release lipase. Makes sense. Bile emulsifies the fat, surrounds those smaller fat droplets so they can't reaggregate into the big fat globules. Keeping them small so the lipases can get in there and break the fats down to fatty acids and monoglycerides. Okay, so that's one pathway here, one flow chart kind of that you need to remember. The other one is S cells. S cells, they actually detect a little bit of acid coming from the stomach. The S cells detect that acid, they secrete secretin. Secretin, again, just like CCK, reduces gastric emptying and then travels to the pancreas to stimulate the pancreas to release bicarbonate. Makes sense. Bicarbonate then neutralizes the acid in the small intestines. Okay, again, bicarbonate is released through CFTR channels. Okay, so I talked about those a long time ago in terms of cystic fibrosis and the transport of chloride through those channels. But chloride and bicarbonate are both anions. CFTR actually transports bicarbonate as well. So the take-home message here is that people that suffer from cystic fibrosis really are compromised in their ability to secrete bicarbonate. So a lot of times it causes a plug of the pancreatic duct. And literally there's a mucus plug. They are sometimes compromised in their ability to secrete all digestive enzymes. Yeah, that's a big problem. So even the mildest form of cystic fibrosis, a lot of times they have pancreatic dysfunction. They have to take pancreatic enzymes as a result. Okay, so this also says that it detects osmolytes, an increase in osmolarity. Also, there's feedback to cause pyloric contractions, again, decreasing gastric emptying, decreasing motility in the stomach, and decreasing hydrochloric acid secretion. Okay, so since you guys are here, remember, you need to know how to label this chart. Start this chart. Okay, this is one that's you'll need to know. Pretty easy though. I forgot to mention that CCK also relaxes the sphincter of Oddi. So CCK not only contracts, causes the gallbladder to contract, increasing the amount of lipases that are released from the pancreas, but also causes the sphincter of Oddi to relax. You can see this is a common duct between the pancreas and the gallbladder. That's the common gateway. There's a common bile and pancreatic duct. Oh, I should have showed you this. Okay, so here's the duct cells. These are the duct cells that express CFTR. That's the channel responsible when it's not there for the disease cystic fibrosis. And these duct cells they express CFTR and they secrete the bicarbonate in the duct. Yeah. Yeah, yep. What increases pyloric contractions? All three of these things. So once you just start getting a little bit of food into the intestines, then the pyloric junction really starts to contract because now it's not about delivering all of the food into the stomach. It's just like piecemealing it into the stomach slowly. Yep. So this is what is known as decrease in gastric emptying. Yeah, you have a question. Yeah.
Wait, so what do we need to know about the chemoreceptor that detects the
Nothing. Okay. Nothing. You do need to know I cells, CCK. You do need to know S cells, secretin. You need to know G cells in the stomach that secrete gastrin. Let's go back to the... So here we go. G cells in the stomach secrete gastrin during the cephalic phase and the gastric phase.
And then what do
They are initiated by neurons. This is gastrin-releasing peptide. So it's mediated through the vagal efferents. Remember the vagus nerve is the parasympathetic. That's the cranial nerve 10. That's the main autonomic nervous system neurons that are mediating the parasympathetic responses. Okay, so it's these vagal efferents that are releasing or causing the release of GRP. So they're pretty much stimulated by the vagus nerve.
Okay, so the G cells don't really have anything to do with the intestinal phase?
No, yeah. I'm just kind of running down some of the cell types that I do want you to know. I cells, S cells, G cells. And then there's one more. There's the GIP. Okay, so that's one we didn't actually talk about yet, GIP. I think this is hilarious because when I was going through graduate school, that was called gastric inhibitory peptide. Gastric inhibitory. So they knew that it actually inhibited gastric emptying. Now it's called glucose-dependent insulinotropic peptide. So they expanded the name. They kept the acronym the same, but now they changed it to glucose-dependent. So it's sensing glucose. Glucose-dependent insulinotropic. It's then initiating the secretion of insulin from the beta islet cells. So now GIP is glucose-dependent insulinotropic peptide. And it gives you an idea of what's happening with the regulation. Detecting sugar, sending a signal to the beta islet cells to release insulin. Makes sense. You just ate a bunch of sugar, and you better get insulin ready to be secreted. Okay, so that's pretty much it. You guys feel pretty comfortable with that? It's really about just memorizing those cell types, what hormone they release, what do they do. Yeah?
So you said that the GIP cells, they're in charge of basically telling the islet cells to release the insulin?
Yep, pretty much.
What's the signal molecule, or is there not?
What's the signal molecule?
Yeah, or is it just the GIP?
Yeah, GIP is the signal. So these cells are called, I should be more specific, they're GIP releasing cells, not real original. GIP releasing cells, they release GIP. That's actually the molecule that travels to the beta islet cells that then triggers the release of insulin. However, you do have to have some blood glucose. You have to have an elevated blood glucose levels for the insulin to actually be released. There's some really cool potassium channels that detect ATP inside the cell. So glucose enters into the cell, makes ATP. It actually binds to these potassium channels, which inhibit them, depolarizing the cell, and then insulin just floods out. It's kind of a pretty cool mechanism, actually, that we didn't really get into. But yeah, great question. So not only is GIP necessary, but you have to have high glucose levels in the blood. Yep. All right, so here, this is the chart, gastrin, CCK, secretin, and GIP. Don't worry about this one. You can just X through it. This is way too much information. But if you're curious, you guys, you can see that there are so many hormones that are involved in the GI tract. I'm just giving you the big four, okay? Gastrin, CCK, secretin, and GIP. Okay, so we're already at 6:30. Okay, so what is important after this? I already talked about the 3 types of motility. In the small intestines, it's segmentation, peristalsis, and migrating myoelectric complex. I can tell you there really isn't any questions related to the structure of the intestinal wall. Let's just be honest. Okay, so here we go. Here's where I do want you guys to know segmentation, peristalsis, and then migrating myoelectric complex. Then I think what's important is how these enzymes are digested, okay? So now we're kind of picking up where the pancreas left off. The pancreas does actually secrete a number of different digestive enzymes. These are different from the hormones. Digestive enzymes include trypsin, chymotrypsin, elastase, and carboxypeptidase. They're kind of listed right here on the chart. These start to break down proteins. Actually, I shouldn't say start, because they start to break down in the stomach by pepsin. Trypsin, chymotrypsin, elastase, and carboxypeptidase, they break down polypeptides into smaller peptides. Lipase, of course, breaks down fat. Amylase, now there's a difference again between salivary amylase and pancreatic amylase. Break down polypeptides. And then you have the ribonucleases that break down nucleic acid. Pretty straightforward, but want to make sure that you guys know the different types of enzymes, and really what we're talking about, the breakdown of proteins, fats, carbohydrates, and nucleic acids. Okay, there's really nothing on enterosymbionts. I even said in class, don't worry about that. Engulfing pinocytosis, phagocytosis. If you guys actually took cell biology, you would go into a lot more detail on those. What I want you guys to know, kind of pick up, is when we start to absorb some of these molecules. So let's start with the absorption of glucose. Remember, these are mediated through those sodium glucose transporters, or SGLT. SGLT. They're located on the apical membrane. This is kind of a nice figure. Actually, this is a really nice figure to show that pancreatic amylase is broken down into, it's an enzyme that breaks down polysaccharides to disaccharides like maltose, to then monosaccharides. And then the monosaccharides are taken up by the cell either by the secondary active transporters, the SGLTs, sodium glucose transporters. They use the sodium gradient, you can see the big Na, little na, using the sodium gradient to move glucose against its concentration gradient. Fructose, though, actually moves into the cell down its concentration gradient by these GLUT transporters. You can see on this other side, these are the GLUT5. GLUT5. You don't need to know that, but you do need to recognize the difference between secondary active transporters and facilitated diffusion. Fructose continues across the basolateral membrane, again by facilitated diffusion, as well as glucose. These are all mediated through these let's see, GLUT2 is the orange ones there, GLUT2. Now, it is interesting to note that there is a difference in the expression levels, whether we're talking about low glucose or high glucose. So you can definitely see an increase in GLUT2 and GLUT5 facilitated those transporters that mediate facilitated diffusion. Oh, let me go back too. Make sure that you guys remember that water is also transported by these SGLT1 transporters. So not only do they transport sodium and glucose, but water is also transported and absorbed in the small intestines by those transporters. Okay. Amino acid, very similar. You can actually see, again, pancreatic proteases like trypsin and chymotrypsin, and carboxypeptidases break down proteins into smaller peptides, and it's these secondary active transporters that then transport amino acids against their concentration gradient using the sodium gradient. Then amino acids are then transported across the basolateral membrane by facilitated diffusion, by these amino acid transporters, okay? Pretty similar to what we saw with the glucose. All right, so then fat. This is a little bit more extensive. I do want you guys to know how to think about bile. Bile is an amphipathic molecule, meaning it has a polar end and a non-polar end. And it helps to emulsify fat. I told you that you guys needed to know the bile formation. Don't really need to know that. So I do need you to know that it is an amphipathic molecule, non-polar side, polar side, and you can actually see the non-polar side faces the fat globule, where the polar side is facing the aqueous solution. So imagine the polar side meeting up with another polar side. Again, it basically does not allow reaggregation. They'll repel. Okay, so bile is very important, not in the breakdown of the big fat globules, because it's really the mechanical digestion that breaks them down into the smaller fat droplets. The bile just surrounds the smaller fat droplets so that they can't reaggregate. Yeah? Yep. You want me to go back to that one? Okay, so the question was with this slide, where is this occurring? This is all still in the small intestines. Yep. I would say the majority of it too is in the upper part, the duodenum and the jejunum, not really the ileum. Okay, so going back, let's talk about fat digestion. The bile helps to emulsify the fats. Then the lipase, this lipase can get into the smaller fat droplets and start to break it down. This is an enzyme that breaks the fat down to monoglycerides and fatty acids. Now they'll toggle back and forth between these micellar formation. These are micelles. They're kind of like plasma membranes in a circle. Okay, so plasma membrane is made up of a lipid bilayer, and the fatty acids face the inner part where the polar sides face the outer part, right? A micelle is just like one part of the plasma membrane in a circle. Okay, so it's only one of the layers that just forms a circle. It's called a micelle. So all of the fatty acid, the hydrophobic components face the middle, and the polar sides face the outside or the aqueous solution. And it'll toggle back and forth between these monoglycerides and fatty acids. Now remember, these hydrophobic molecules can easily just move across the plasma membrane. They don't need any kind of protein to help. So they easily kind of move across the apical membrane. This is how you absorb fat, where it's then actually repackaged. I think that's kind of interesting. It's repackaged back. The monoglycerides and fatty acids are repackaged to triglycerides. And then you can kind of see here the triglycerides are made up of the fatty acid chain and the glycerol backbone. Then you add a protein, and that's what's called chylomicrons. It's the triglycerides plus this protein. Okay, so then now they're called chylomicrons, chylomicron assembly, and then it's transported by exocytosis into the lacteal or lymph vessels, where ultimately it ends up in adipose tissue, right? Adipose tissue. Okay, know that bile is recycled as well. So your liver produces the bile, stores it in the gallbladder, delivered into the small intestines, where it's then actively taken back up, these bile salts, and delivered back to the liver by the hepatic portal vein. So it's 95% of bile is recycled. Only about 5% of bile is lost in the feces. All right, you guys, this is pretty much just a flow chart reviewing CCK because of bile. Secretory, there's differences between the diarrhea too. Can't forget about diarrhea. All right, so secretory diarrhea, the classic example is cholera, where you get the GS pathway stuck on the on position. It literally binds to adenylyl cyclase, and you just keep getting increase in cyclic AMP. It really increases cyclic AMP levels within the cell, which enhances chloride secretion. Water's going to follow, and it just creates a profound secretory diarrhea. Again, with cholera, you can lose all of your bodily fluids in a 24-hour period. Osmotic diarrhea occurs, and this is a good test question too. It occurs when you have enzymes like lactase that aren't working properly. So if you're lactose intolerant, it's because you don't either have lactase or your lactase isn't working properly. So what happens? You don't get breakdown of lactose. It stays in your intestines and acts as an osmolate, increasing the osmotic driving force for the movement of water to enter into the small intestines, causing a severe diarrhea. That could be any enzyme not working properly, lipase, lactase. There's a number of things. If these enzymes are not working, then that'll cause an osmotic diarrhea. Malabsorptive and motility related are really types of osmotic diarrheas. Malabsorptive just means that there's inflammation of those epithelial cells, which compromises its ability to absorb these nutrients. Again, glucose stays in the small intestines and acts as an osmolyte. Water, it'll increase the driving force for the movement of water to enter into the intestines and cause diarrhea. Motility related, again, because maybe you're a runner, this is what I think of, runner's diarrhea. The motility of the gut is too fast, so it doesn't allow for sufficient digestion. Then you have undigested material in the small intestines, again acting as an osmolyte. Water's going to enter. There's going to, again, increase that osmotic driving force for the movement of water, and it'll cause a severe diarrhea. Dysentery is when you just have diarrhea with blood. Ugh, that sounds gross. But that's what dysentery is. All right, so moving pretty quickly, the colon. I would say know what hostra is. Hostra are these pouches. Hostration is when these pouches actually literally they just mix. And they actually constrict independently of each other. So they one pouch constricts, then the other one. And really what it's doing is it's forming feces, like big mud pies. That's a good visual, right? All right, forming feces. Mass movement is when you get a simultaneous constriction in the ascending colon, and at the very same time, the transverse colon relaxes, pushing that feces down the line. So a few more things, two more things. Defecation reflex. Defecation reflex I can't say this enough. Defecation reflex is initiated by stretch in the rectum. That's probably the most important thing of this whole process. So remember those combs that I was talking about in the stomach? These comb symbols are mechanoreceptors. So they're detecting stretch or distension. So as soon as you get stretch in the rectum, this is a true reflex. Sends a signal just to the spinal cord. Not all the way to the central nervous system, just to the spinal cord and back. Causing movement in the sigmoid colon. A relaxation of the internal anal sphincter. And an immediate constriction of the external. That's where you like, oh my gosh, I better go to the bathroom right now, right? You literally kind of constrict that as you waddle down the hallway. I'm getting tired, you guys, getting punchy. Okay, so you get the constriction of the external anal sphincter, but see, this is actually under skeletal muscle control. So you have the ability here. This is voluntary. This is from the cerebral cortex. You can voluntarily relax that when you go, number 2. Okay? You actually relax that external anal sphincter. Finally, ghrelin, leptin, and peptide YY. Don't worry about this crazy figure here. You're not going to have to know neuropeptide Y releasing neuron and pro-opiomelanocortin. I would say, just to make it simple, you guys traveled all the way here. Ghrelin increases appetite. It's released from the stomach. Leptin decreases appetite. It's released from white adipose tissue. Like I said in class, it was supposed to be the diet pill, till they realized it just increased the amount of white adipose tissue or fat tissue. And then peptide YY from the colon also decreases. All right, that's it. Don't worry about this one. We actually covered it today. Glucagon, insulin. Okay? All right, so what do we have? Anybody want to go through? Whoa. Am I still recording? Yes. Am I still recording? Hello. Okay, this timer's still going. What does this mean? If I hit OK, will it stop? No. Oh, they're just... Oh. Okay, I'm just going to leave it because I don't want it. It still looks like it's recording. I know, right? It stresses people out. You're not the only one. I know. What's that? I did. I cleaned it. I did clean it off. You guys know Chris Falk? He's another professor. He's like, that really stresses me out. When I went to a meeting, he's like, what? Okay. I think that I'm going to leave it on this one right here. I think one way to do this is it might be worth your while to actually kind of draw on the board. Do you guys, or do you think it's just easier to walk through it? Draw on the board? Because then you can see where it's coming from. I can actually, let's see here. I'm going to draw on the board. Yeah. Oh, wow. Ah, yikes. Ah. Wake up. Wake up here. Okay. So, I think it's easy. Let me just turn this off. Okay. So, if you kind of know what hormones are coming from the hypothalamus, then traveling to, we're going to say the anterior, posterior, pituitary, and then target organ. And then various target cells. I think sometimes it's easier to put it all together now, okay? So let's start. Do you guys want to start with the stress response? We'll start with the stress response. Okay, so from the hypothalamus, this is corticotropin releasing hormone. Okay, this is also called CRH. It travels to the anterior pituitary, where it releases ACTH, which is adrenocorticotropic hormone, or ACTH. Okay? So, corticotropin releasing hormone comes from the hypothalamus. It travels down those portal vessels to the anterior pituitary, where it stimulates and stimulates the secretion of ACTH. That travels to where? Adrenal gland. And what does it stimulate? Cortisol. Glucocorticoids. Glucocorticoids like cortisol. In this case, too, you don't have to know whether this is the zona fasciculata. Don't worry about that. Adrenal gland is just fine. So now, this glucocorticoids, what are some of the things that it does? It increases what?
I'm going to guess blood pressure.
Oh, I could see that happening, but that's more epinephrine. Vasoconstriction. But it does increase blood sugar. It promotes the breakdown of fats and proteins. Promotes fat and protein catabolism. And the big one, why does everybody get sick during finals week? Suppresses the immune system. And this is pretty much why it works as an anti-inflammatory too, right? Why do people get cortisol shots when they have carpal tunnel syndrome or, knee issues or foot issues? It suppresses the immune system and the inflammatory response. So this is the kind of thing that I just want you guys to put together, because the questions are going to be very straightforward. It's pretty much, where does this hormone come from? What does it do? Okay. Okay, let's do the next one. Thyroid. Okay, so thyrotropin releasing hormone. Ooh, spelled that wrong. Or TRH. Comes from the hypothalamus, travels to the anterior pituitary, where it releases thyroid stimulating hormone. Or TSH. Okay? That hormone then travels to the thyroid gland. Thyroid. Where the thyroid now releases T4. T3, triiodotyrosine, and thyroxine. T4 is thyroxine. I think Synthroid is the commercial brand for thyroxine. So if you had some kind of thyroid cancer, they might destroy your thyroid, remove it, and you'd have to take a lifetime. For the rest of your life, you'd have to take thyroxine, Synthroid. Okay, so it travels to the thyroid, where the thyroid now produces T3 and T4. That's the big question. What does T3 and T4 do? Yep, increases epinephrine. Oh, actually what it does is it sensitizes the cells to epinephrine. So now the cells actually are more sensitive to epinephrine. So it sensitizes cells to epinephrine. What does that do? It increases cellular respiration. Increases oxygen consumption. And ultimately increases metabolism, right? So, what are some of the diseases associated with that? That would be hypo or hyperthyroidism. Remember, hypo means too little, hypothyroidism. And hyperthyroidism, too much, excess of T3, T4. We didn't talk about some of the diseases associated with the stress response. In this case, we talked about Cushing's disease. Remember the horse, pot-belly appearance, laminitis. I think even in humans, they have a rounder trunk, tiny legs because of the muscle wasting. And then they call it a buffalo hump. You usually get fat deposits in the middle of your scapula. So there's very, very definite features or occurrences that happen with Cushing's disease. Okay, so that's part of the stress response. Okay, you guys good with all that? I kind of went back a little bit. All right, so can I erase all this? You guys pretty good? Or maybe, no, I'll erase it. Okay. This is kind of what that worksheet was all about. Okay, so let's do the sex hormones. Hypothalamus, gonadotropin releasing hormone. Gonadotropin releasing hormone. That triggers the anterior pituitary to release follicle stimulating hormone. Or FSH. And this is in the anterior pituitary. And LH, which is luteinizing hormone. Is it EIN? Can anybody tell me if I spelled that right? It's kind of a weird spelling. Luteinizing hormone. Okay, so the target organ for males and females are going to be different. follicle stimulating hormone is actually going to travel for males to the testes. Oops. Where it's going to initiate spermatogenesis. And I believe this is in the Sertoli cells. We didn't even go through that in class. Sertoli cells. In females, FSH travels to the ovaries, where it's responsible for follicle development. So every month you have follicle development that leads to a maturation of the ovum. Okay? And then luteinizing hormone for males travels to the Leydig cells to produce testosterone, androgens. Okay. In females, a surge of LH is responsible for ovulation. Luteinizing hormone. A surge of LH. I mean, it is literally a spike right before you ovulate. Surge of LH causes ovulation. You guys pretty good with that? Okay, the last two of the anterior pituitary is prolactin and growth hormone. Okay, so these are funny. The hypothalamus is not very original, but it's growth hormone releasing hormone. And for prolactin, it's prolactin releasing hormone. Okay, so growth hormone releasing hormone and prolactin releasing hormone. Okay, so let's go to the growth hormone first. Growth hormone releasing hormone is released from the hypothalamus, travels to the anterior pituitary, where it releases growth hormone. Now, where does the growth hormone go? This is a good one. Growth hormone actually travels to the liver, where the liver actually secretes insulin-like growth factor. Insulin-like growth factor. It's actually IGF-1. And then it's IGF-1 that travels to various cell types, where it initiates growth of specifically muscle and cartilage. Cartilage and bone. Oh, I spelled cartilage wrong. And bone. So some of the diseases are pituitary dwarfism, lack of growth hormone. Pituitary dwarfism. Gigantism. Can't spell anymore. Gigantism and acromegaly. So pituitary dwarfism is when you have a lack of growth hormone. Gigantism is when you have an excess of growth hormone before puberty or right at puberty. Acromegaly is when you have an excess of growth hormone after puberty. So gigantism is caused usually by a pituitary tumor, and it causes people to become really large in size, like we talked about Andre the Giant. If you get an excess of growth hormone after puberty, it usually just causes the mandible and the cranium to become really thickened. So it's very distinct from gigantism. And then prolactin, let's go to prolactin. Releasing hormone releases prolactin. Oops. From the anterior pituitary. Now, I just want to make sure that you guys realize that this is very distinct from oxytocin. Prolactin travels to mammary glands. Where it stimulates the development of mammary glands and the production of milk. Stimulates development and production of milk. This is different from oxytocin. Okay, so oxytocin is responsible for the ejection, the letdown and the ejection of milk. Okay, so one thing I didn't concentrate on in the class, but I really need you guys to know this too, is that vasopressin and oxytocin are secreted from those neurosecretory cells of the posterior pituitary. You guys know all about vasopressin now. Vasopressin inserts those water channels in the collecting duct and promotes water reabsorption. Oxytocin is responsible for the ejection of milk. It's also very important in labor. Remember that classic positive feedback mechanism? When a woman is in labor, basically what happens is you get stretch of the cervix and contraction of the uterus during labor. It feeds back to the posterior pituitary. You get more oxytocin release. That causes more uterine contractions. And then it's just like a positive feedback mechanism until a major event like birth happens. So, okay, oxytocin, vasopressin, coming from the posterior pituitary. Yes?
Can you repeat what you said about vasopressin?
Yep. Vasopressin is also called anti-diuretic hormone. And it is released from the posterior pituitary in response to high osmolarity in the blood. Okay, so you've got a lot of solutes in the blood, maybe because you're severely dehydrated. It's going to be detected by the hypothalamus, which is going to produce and secrete vasopressin from the posterior pituitary. Then it travels to the kidney, specifically the collecting duct, where it triggers vasopressin receptors. It's a GS-coupled receptor that then inserts water channels or aquaporin channels into the apical membrane, promoting water reabsorption so that you can produce a really concentrated urine. That's why it's called anti-diuretic hormone. You guys remember that? Yeah.
So the only one of everything we just went over, the only posterior was the prolactin and I'm sorry, not the prolactin, the oxytocin, the vasopressin.
Yes. The only ones coming from the posterior pituitary are vasopressin and oxytocin. Okay, so the 6 hormones, there's more. We didn't talk about MSH, melanocyte-stimulating hormone. But the main ones from the anterior pituitary are TSH, ACTH, FSH, LH, prolactin, and growth hormone. Those are the 6. And then we pretty much covered today, we covered a lot of glucocorticoids, the adrenal glands. Do you guys need me to go through thyroid? Are you exhausted? That's a lot of physiology, isn't it? I think that we've covered just recently some of those other hormones. Like I said, this exam is pretty straightforward. So if you can actually just kind of write out what we did, put it all together in one big map, then you won't have any trouble on the exam. Okay, so it's important to know where the hormones are coming from, what is their target organ, what is their ultimate function. If you can just kind of write it all out, shouldn't have any trouble at all. What are some of the diseases associated with these? Like we talked about pituitary dwarfism, gigantism, acromegaly. We also, for insulin and glucagon, diabetes mellitus, no type 1, type 2. Then we talked about parathyroid hormone. We didn't really talk about the disease associated with it, but that would be bone loss, osteoporosis. Okay? And I think that's it, you guys. Thyroid stimulating hormone we did talk about. Yes?
We don't have to worry about any math calculations.
No, there's not an equation sheet. I've had a lot of students come up to me and say, oh, I really like the equations. Believe it or not. No. No. Yeah, no calculator, no equation sheet. This is a little different than what the exams you've had in the past. I think it's kind of more like zoology in that it's more memorization. It's more like making flashcards and just knowing hormones. I think the other 3 there's more of a logic, like a reasoning component. This is just straight rote memory, right? Straight up. Just memorizing stuff. I'm exhausted. You guys must be exhausted. My 8:30 class must be exhausted, you guys. Yeah. You've had a long day. It looks like a 12-hour day. All right. Any other questions? I can stick around for a few minutes, you guys. Yes. Yeah. Oh, sure. Yeah, that was it. And that was one thing. Calcium and parathyroid. They're basically the yin and yang, right? They have their antagonistic pairs. So the thing to remember is that your thyroid hormone both calcium and it causes the osteoclast to go by and calcitonin is the exact opposite. It protects eye calcium. How are you doing that? I've noticed, yeah. So, yeah. 3 to 5 in this room. Okay. Yeah, in this room, 3 to 5. I'll see you tomorrow. Yep.
If we didn't do about the bottom, I don't have that we were. Oh, you're here tomorrow. Can we still come and take tomorrow? Okay.
Yeah. If you feel pretty good about it, yeah, I would say
And then if we want to remix,
yeah. Do that still. I was thinking about that. I think it was like across the board, 20, everybody else they were going to take. It was pretty sweet. So I was going to do like
you complained that it's broken.
So I don't know if you
Okay.
all of the 7 of you decided on one exam, so I don't know. Okay, so I think there's only about 2, and I'll just bring all 3.
It was yeah, and so if we do retake one, is it the average or do you just check?
I don't average it. I don't curve it.
Yeah.
So, yeah.
Like you don't add like the first or the second one, but you know what I mean? Like you don't take the first score.
Oh,
And
no.
then the second score you
No,
add
I
it
just
divided by
take the best
2.
of the 2.
Okay.
If you get 100 on this one.
So say I hear words, then I get my first.
Yeah.
Okay.
It's a very low risk.
Okay, I just like
Yeah, definitely.
okay, thank you.
You're welcome. Yeah.
Right?
Yeah.
And my grandpa was still pulling him in the leg and not realizing his little brother.
Cute brother. Okay.
I mean,
Or 11
technically it's kid. But
I just have TAs. We have picture press for an hour. And then I have to run off in a minute. So I'm only going to be here a few hours. But I will be here all day tomorrow. Yeah. You're taking the life hack.
I was like, wait, this is it. And I never.
Yeah, that works. Yeah. Yeah. I will just hold off and give you a grade when you're ready. Oh, yeah. No big deal. Not at all.
Okay.
Yeah, just get it to me when you can.
All right.
Yeah.
Thanks.
You're welcome. So it's like we're saying that
Tomorrow.
tomorrow.
Yeah,
Oh,
3 to
not
5.
today, yeah. But
So if you wanted to take if you wanted to take the lecture
Yeah.
final early and the makeup,
oh, yeah.
3 to 5 in this room tomorrow.
Yeah. And then it would just be like regular semifinals, like or like intercut stuff. Like,
Yeah, yeah. I think that I am going to have you guys do the Scantron. So
got it.
yeah, just bring the number 2 pencils. Oh yeah, got it. Thanks. Absolutely. Love to see that. Yeah, absolutely. When do you need it? I always have to put a due date for myself. Not okay. Just I know you well now, so I will recognize your email and just I will respond. But when it gets closer, I always ask you just remind me. I don't mind. Some professors get really touchy about that. Yeah. Oh, I know exactly. But I like the reminders. That always I will reorganize myself.
Sure. And should I just send you an email then?
Yeah. Yeah. Okay. That sounds great. So is it med school? Nice. Yeah. Okay. Sure. I'd love to do that.
Thank you so much.
Yeah, yeah. I'll be rooting for you. I have my fingers crossed for you.
Thank you.
All right. Yahoo! Yeah, you too. I hope that helped. Good.
Thanks for a great semester.
Oh, thank you. Thanks for saying that. That was really sweet. Oh, I am so glad. I really enjoyed you guys too. You guys were so fun, really amazing.
It was like taking biochemistry was very, very boring, but it was really nice that I took it on the same time as this because it helped me understand this better.
Oh, nice.
It made me actually really, really enjoy physiology.
Oh, that's so great. Yeah, you could apply kind of what you were probably learning, and yeah, that's awesome.
It was like I had common, like I'd already understand like kind of what was going on, so then like seeing it actually in the body was really cool.
Oh, that is really cool. Yes. Oh, well, thank you for saying that. I love hearing that. Have a good evening.